So this talk is about genetic engineering for fun and profit. It's not in? It's not. Okay. Okay. Can everyone hear me now? Okay, cool. So this is about genetic engineering and as a biohacker or with it kind of, hold on. Get it back? Okay. I think this is not a good connection. Yeah, I have one in my. Okay.
Magic spot.
Okay. I think I have one in my backpack. I have an adapter just like that. Yeah? Oh. Should I get the other? Okay. Well, we'll use this for now then. Okay. So, why would we do citizen science and DIY bio? Oh, sorry. So in academia, you can do science and bioscience, but the thing with that is that your principal investigator, who's some professor with a lot of experience, kind of directs what you're allowed to do. So if you want to do your own thing, you really can't without convincing other people. And even the PI had to get a grant or something to do the research that they're doing. So the other route is to, get venture capital or, get a corporation to kind of buy into your idea. But for that, the idea has to be something that you can convince other people that it's profitable or worthwhile doing that type of research. And so basically, if you're just curious about biology or just want to just do research for the sake of research, you really have to do it yourself. And that's kind of how the citizen science biohacking came about. So the thing is, currently to develop a new drug, it's $2 billion just to do all the government regulatory stuff. So corporations are not doing research anymore on like novel antibiotics or novel ways of treating a disease. They're kind of taking the safe route, and they only want to develop drugs based on existing things that they know work. So there's a space for citizen scientists to kind of research new concepts and new ideas. So that's a reason for why you might want to do citizen science. And yeah, also as hackers, the hacker ethics exist, which I think our culture has, which is the idea of sharing, openness, decentralization, making the world a better place. So, DIY bio can definitely play a role in helping that. So the types of things that DIY biologists can do, or biohackers, is we can modify bacteria, fungi, or plants and get them to produce proteins or even molecular devices, molecular machines even, or even do computation. There's been work done on getting computer gates like AND and NOT gates to work inside cells. So the field of synthetic biology is kind of growing because of these abilities. The other thing is you can find new species out there that may be undiscovered because, there's some mushroom growing somewhere and nobody's actually looked at it to see if it's a new species, because a lot of them look the same. And what I found out is that you can actually find new species of mushrooms quite easily, because they're like everywhere and they haven't all been cataloged. So there are people who are doing that, and they're finding new species all the time. The other thing you can do is, and there was a group that did this in New York, that they went to restaurants to check whether they were actually getting the fish that they thought they were getting. And it turned out that in a lot of cases it wasn't. So maybe it wasn't. If it didn't taste like salmon, maybe it wasn't. So the other thing is you can search for, make new discoveries, or, find new antibiotics even. And it's actually quite easy to, get a sample of something and see whether it has antibiotic properties. The other thing is negative results. In academia or in the publishing world, they don't want to hear that, oh, something didn't work. But in the DIY bio, I found that, people want to know things that don't work, so they don't end up trying all this stuff and then it doesn't work. So that's one thing that's kind of more present in, in the biohacker community. So what kinds of things we can't do is like create a zombie virus. It's actually very difficult to create a zombie virus, and there are all these enemy countries that are trying, and they haven't been able to do it because the properties that you need to have in a complex virus like that is very difficult. And nature is actually also trying to make a perfect virus through evolution, and it hasn't been able to do it because our immune systems are actually quite good in the way it's set up, that our populations and all have like diverse mechanisms of immunity. So there's no disease actually still found that that all humans are susceptible to. So like even HIV, there are people with mutations that aren't affected by it. So the other thing is, it shouldn't be overhyped that some biohacker is going to come up with a cure for cancer or some disease, because some of those are overpromises, and so you shouldn't necessarily buy into that stuff. But you can definitely do good research. The other thing is, you do need to follow the law and be ethical if you're doing any type of bio research. And there are basic biosafety principles, like Keone mentioned in the previous talk, like you don't work with any dangerous pathogens, and you also make sure that the lab that you're working in is fully compliant with the biosafety level that are out there. So if you're just working with bacteria, you can do it in a BSL level 1 lab. That's, when I say bacteria, I mean like safe bacteria that are like E. coli. E. coli is pretty common, and that you can do it in a BSL level 1 lab because E. coli is actually found everywhere. So it's not going to be dangerous to play with that. And then if you want to do stuff with like mammalian cells, then you have to go up to BSL level 2, because mammalian cells can actually act as a host to viruses that affect humans. So if you have a sample somewhere and you sneeze on it, you can put the cold virus in that sample, and then it can like spread. So that's why you need to have extra precautions if you're working with mammalian cells. So what is genetic engineering exactly? It's basically we're modifying the DNA inside an organism to give it new properties that we want, whether it's resistance to pesticides, able to survive drought conditions. And actually this plant there, this is actually what corn comes from. Before it wasn't genetically modified on the DNA level, but basically the pre-Aztec civilizations in Mexico, they took this plant and did artificial selection, breeding, and hybridization with other similar species, and they developed corn, which looks completely different from this plant. And this plant is called teosinte. So that's not really so. So the next step beyond this is happening now with GMO corn, is to actually insert genes specifically that we want to have in it. Instead of, when you do a hybridization or even artificial selection, you're not being very specific, and you're like expecting nature to do what you want, and that's kind of difficult. So what is DNA exactly? It's basically the protein encoding system of life, and every cell contains DNA, and the DNA is used to make all the proteins that a cell needs, and when it divides, it makes a copy of its DNA and gives it to its offspring or next generation. So DNA makes proteins, and the proteins you can kind of compare it to like Lego blocks, in which you can build molecular machines out of it. And these proteins are a chain of molecules called amino acids, and these amino acids have different properties. So you can build a structure with that. That structure is going to have certain properties, like the ability to bind to something or fit into something. And it can also be used as a machine that can process other chemicals that are in the cell. So a little more detail about what the DNA strand consists of. So there are these nuclear-based molecules that form a duplex structure of basically two strands that contain the four base molecules of DNA, which are A, T, C, and G. And the sequence at which they exist define the properties that the protein that it's going to make will have. And it also specifies how much of the protein is going to be made and when, because there are things called polymerases which kind of act on the DNA and copy it or make or help in making the proteins from it. So each 3 letters of DNA specify one of 23 different amino acids. So these amino acids, like one example is glycine, which is flexible. So if we want to connect 2 proteins together, we can make a linker out of glycine and attach the 2 proteins so that they won't affect each other. So one reason you might want to do that is if you have if you want to tag a protein with fluorescence without affecting its property, but you want to see where that protein is going or what it's doing, you can attach it with a linker. And then within these DNA, we can see patterns in there that are repeated through nature. For example, like if we recognize a specific sequence, we can tell that it might be forming a structure called a beta hairpin, or HNH, which is an endonuclease that cuts DNA. So if you're looking at a DNA sequence and you look at the corresponding amino acid sequence, you can actually analyze it and kind of guess at what this protein might be doing by looking at those motifs. The other thing is that DNA has this phosphate backbone, which kind of provides it with a negative charge. And because of that, we can actually separate out DNA by strand length by having it run across a gel. So if you see up here, we load samples of DNA, and have an electric current, positive over here, and the DNA will slowly move towards this. And if the DNA strand is long, it'll move slower than if the DNA strand is short. So we can see here, this is basically a shorter strand than the one down here. And this one consists of multiple strands, and the shorter strand is up here. And that has a number of different uses too. The other thing we want to do with DNA is to find out the sequence of it, to identify whether what a particular gene is, or whether a particular sequence exists in an organism. Currently, this costs about $1,000 to sequence. About $3,000 to sequence a full human. But in the next few years, it's going to drop below $1,000, and then maybe even below $100 by 2025. That'll open up a lot of possibilities. So basically, it's cost pennies to figure out a DNA sequence of something. The other thing is that we can make DNA ourselves. Companies are out there that can do it, and they cost about 30 cents per base pair. So each letter of the DNA that you need made, is very cheap. So you can get a long strand of maybe even a full gene for like under $100. So what we want to do is basically modify the organism by interacting with its cells. So we put the DNA inside the cell of the organism, and these cells, you can have single cell organisms like bacteria, or you can have multicellular organisms like people or plants. And they all use DNA, and they all follow the same code. So a particular sequence of DNA will always produce the same amino acid chain, regardless of if it's in bacteria or in humans. So that's a very cool property of DNA that is very helpful. And when we modify cells, we can get them to do stuff, and we can basically hack them. We can hack their signaling pathways. We can hack how they communicate with each other, and even hook into their signals to make their signals act the way, or activate things that we want to activate. One type of molecular machine that's inside cells is called a polymerase. And this polymerase is attracted to certain DNA sequences. So when we can actually put our own sequence of DNA inside the cell and use that promoter sequence to get our DNA to be expressed. And so when you have a thing called a plasmid, you make sure it has a promoter on it so that the protein gets read. And the other thing is these promoters can be activated only in the presence of some other substance. So you can activate it at will by providing the cell with a particular chemical, and that'll activate the promoter and perform the function. And so a cool thing is that the cells themselves contain machines that we can actually use for our purposes. So for example, restriction enzymes, they cut DNA at a specific sequence. And so the bacteria's own DNA doesn't have the sequence that the restriction enzyme will cut. But if it's infected with a virus, it can cut that virus's DNA because it has this restriction enzyme that'll just cut it up, and then it's protected. The other thing that protects cells is the cell membrane. And, the cell membrane is sort of like a filter to only allow certain molecules in. The other thing that cells do is DNA repair and error correction. When the DNA breaks, the cells actually can repair that And we can actually use that mechanism to put in our own pieces of DNA into the cell's genome. So the other thing that that bacteria like to do is transformation, which is they like to take DNA from their environment and actually express it. And see, it's a type of survival mechanism in that they want to gain, because sometimes other bacteria that was in their environment might have resistance to something that the bacteria might find useful. So it basically likes to click on everything and see what it's used for, or whether it's useful. And that's also a good thing because that allows us to shove in our DNA that we want the bacteria to use. The other thing is it has this thing called CRISPR-Cas9, which you can basically use that to cut a specific sequence of DNA that you specify. And it uses that to detect whether a phage that it had seen before whether it encounters it again. It allows it to adapt to that phage so that next time it instantly attacks it. So to do genetic modification, we synthesize a strand of DNA that specifies the code that we want. Then we insert the strand into a cell. The cell will make copies of that strand that will stay within the cell whenever it replicates. So that way you can produce a large quantity. So if you wanted to do this, the first thing you would do is order some DNA. But I'll just show you kind of what that process might involve. So you can go on this website called NCBI, which contains the DNA sequence of many organisms out there. So right here, I've specified this Ranella luciferin, which basically helps sea urchins glow. So it's like it provides bioluminescence. So when we search for that, the sequence for it comes up here. And we can just copy this sequence literally and paste it into a site a company that will synthesize the DNA for us. So we just paste it in there. And then the next step, it makes you promise that it's not a toxin or viral DNA. And it also, but it also does its check to, it has a database of known viral sequences that it checks for and toxins that, you have a question? That's, I don't know. I don't know if they have something, Monsanto patent checking tool or something, but it basically the only questions it asks are: is it a toxin? Is it viral sequence? And it also checks. It doesn't just believe you. It also checks. So, but yeah, I don't know about whether it checks for whether is this a patented sequence. I doubt it though. So the thing is, in DIY bio, we like to use bacteria for creating proteins because they grow very easily, and their chromosome is not protected by a nucleus. So when you insert stuff in there and you want it to get into the chromosome, it's fairly easy in comparison to eukaryotic cells. The only disadvantage is that the proteins are not modified after they're created the same way that they're modified inside cells of complex organisms. So, but the bacteria are pretty awesome because they actually have a lot of enzymes and things that we can use. So some of the technology we stole from bacteria is this idea of plasmids, which they exchange DNA material amongst each other through this plasmid. And by studying the plasmids in the 50s, it was kind of figured out that what you need to have in a ring of DNA, an important thing is called the origin of replication that tells the bacteria cells machines to make copies of this DNA strand. So by modifying this sequence, you can play around with how many copies of the plasmid are going to exist inside the bacteria, and whether it's copied or how many of those copies are there. So that's useful if you want to regulate how much protein you're producing. The other thing you need to have is a promoter, which is what the bacterial polymerase will attach to produce the gene. So we have a promoter here, and this promoter is, in this particular plasmid, activated by a sugar called arabinose. So when in the presence of arabinose, the structure of the polymerase changes so that it gets a binding affinity to this sequence of DNA, this particular coding sequence. It's a short sequence. And then the polymerase attaches there and then starts making the gene. Basically, it copies the gene from DNA to RNA, and then the RNA is turned into a protein. So to do transformation, we basically create a plasmid containing the gene of the protein we want. Like, it could be like banana flavor. Insulin is made this way, actually. You can design a biosensor that can be produced in the bacteria. And the other thing that we want to include in our plasmid is resistance to an antibiotic drug. So this one is ampicillin. And what that does is it allows us to separate the bacteria that took our plasmid from the ones that weren't. So when we add an antibiotic there, all the bacteria that didn't take our plasmid into it get killed. So the only ones that survive have our antibiotic. And we only use antibiotics that are not commonly used in society. So like chloramphenicol is an antibiotic that used to be used in, I guess, the 60s, but it's no longer prescribed. So we don't have the issue of if this bacteria gets out, what's going to happen. We can use electricity or chemicals to basically alter the cell wall or modify the charge on the cell wall so that the cell membrane, so that pores kind of open up, and that allows DNA to slip in. You can do that with electricity or chemicals, even ultrasound works. And you can use like a jewelry cleaner ultrasonic box to do that. And so you can do it very cheaply. So this kind of shows, once you've done the transformation, you get this you have a small Petri dish that you've poured the mix on, and you'll see little dots, which are colonies of bacteria. And you can just pick one of the colonies and put it into liquid growth media. And then you put it into this incubator, which basically shakes it at the temperature that the bacteria like to live. So for E. coli, it's 37 degrees. So eukaryotic cells, their DNA is protected by a nucleus. So you have to do so it's a bit harder to work with it, but it's not that much harder. The other cool thing about the eukaryotic cells is that they modify the protein after it's made, so the proteins can have, I guess, more complex features. So we can also modify plant genes, and there's actually a bacteria called Agrobacterium tumefaciens that has evolved to be able to do this. So it contains a plasmid in it already that when it infects a plant, it shoots this plasmid into the plant so that the plant produces the nutrients that the bacteria need to survive. So what we can do is basically take this plasmid and cut out some of its genes and put in the genes that we want to put into the plant. Then we put this plasmid into the agrobacterium, and the agrobacterium will infect the plant and insert this plasmid into it. So this left border flanking regions kind of help help the gene cassette kind of get inserted into the plant. The only thing is it's fairly random because this gene could end up anywhere in the genome of the plant. So there are some issues with that. Is there a question? So it actually injects it, and so it's a bacteria, not a virus. So it sits on the plant surface and creates injects, like dissolves the cell wall or modifies it slightly so that it can shoot things into it. So it injects this plasmid into it. So it doesn't need reverse transcriptase because this is an actual strand of DNA already. So once it's in the plant cell, the plant cell will copy it because it has this origin of replication. So the plant cell will think, oh, I need to make copies of this. It'll make a bunch of copies of this, and then it'll also incorporate this region into its genome. So a new tool that's emerged in the last few years is CRISPR-Cas9, which allows us to break DNA in a specific location. And then when it's broken in a specific location, if we have another strand of DNA floating around with a similar flanking region on the side, the cell is going to think it's part of its own DNA got broken, and it'll basically try to repair itself by inserting the gene that you introduced into the cell. So it's a very useful tool for making targeted genomic modifications. And the cool thing is that you can buy this protein for $65 from AdGene. I think we're kind of out of time. I actually had much more stuff to go. 5 minutes? Okay. So PCR, I think Keone kind of talked about this, where if you have a small amount of DNA and you want to make large copies of a segment of that in a large quantity, what you do is you create a thing called a primer, which is a short DNA sequence that exists near the segment that you want to make copies of, and you basically use that with a thing called with a polymerase. A polymerase is just something that uses a template of DNA or RNA and makes copies of that, either DNA or RNA. So basically, you use this polymerase with the primer. It'll separate the strands and make copies, and you'll have a large quantity of it. So you can then use that to detect. It's used in forensics, for example. The FBI will use it to see if a crime scene contains a person's DNA. So the FBI actually has 13 markers in the human genome that they look for, which are short tandem repeats. So it's like 4 letters of DNA, but they're repeated like hundreds of times in a particular region. So these are chromosomes, and these are the locations. So if you could modify your DNA and change this location, and if you're in this database, you would be able to get past that. Or something you could do is create small, I don't know if this would work, so if it works and you get arrested, you could create your own DNA. You can make DNA that contains the short tandem repeat sequence of someone that you don't like, and put that at a crime scene. And when they sequence that, when they look for these markers, they'll see DNA with these markers around there. And I don't know if that'll really work properly, but that's a possibility. Yeah, so this is kind of when they separate by strand, so those tandem repeats will be different for each person. So if you run it on this gel, each person at the specified location will have a different number of repeats, and you can look at it with a gel. A way of finding novel antibiotics, you can basically spread bacteria on a Petri dish and then put a small drop of leaf extract or some substance you found that you want to see if it's antibiotic, you can test it that way. Another thing is you can make milk proteins without cows in using yeast. So you basically take the gene sequence of a cow, synthesize that, make a plasmid, put it into the yeast, and the yeast will produce milk proteins. So, and here are some resources for citizen science. Arcturus BioCloud, Genome Compiler, the Odin, you can get good supplies there. Keone talked about them. Synbiota, Benchling has some good software tools. IndieBio actually sponsors people with good ideas. They give out like $250,000 if you have a good company that you want to develop. Yeah, I can make them available. Yeah. So ordering stuff, sorry, adgene.org contains plasmids already, and there's thousands of plasmids that they have that contain fluorescent proteins, proteins that do anything that you can imagine. Well, almost anything. So adgene.org is a very good site. The Odin is actually a good site for biohackers because it's kind of specific for getting you up to speed on DIY bio. For buying things like restriction enzymes, there's New England Biolabs. I just put it as NEB, but it's New England Biolabs. For doing bacterial transformation, you can get the kits from Clone Tech, the Odin, Zymo Research. Reagents you can buy from this location here. DNA, GenScript, IDT. So I kind of give a little plug for, I know the owner there, and he's a nice guy. Okay, and here are some hacker spaces that you can join, and they can help you with doing this stuff. So, yeah, that's kind of, hopefully will give you an intro to getting started with this stuff. Okay. Yeah. Yeah. Okay, any? You cannot necessarily test it. Yeah. Well, yeah. Because it might be doing something bad. You have to go through a FDA. So any other questions? Or I think we're... Pardon? Yeah. Is that just data sets? So, well, there's actually the Panday, no, is it Panday? There's a data set from a professor at Stanford, and now he's at UCSF. His name is Atul Butte, B-U-T-T-E. First name A-T-U-L. And he's got a lot of data that you can mine through and find, especially for cancer. Yeah, and okay. Question? Oh yeah, Hive Bio, yes, they're very good ones. Yeah. Okay. Okay. Let me just put these. There. I just put it up there now. Yeah. Probably nothing, or maybe a huge difference. Well, like something like GFP, you might get lucky and make it brighter than it already is. And there are versions of green fluorescent protein that you found in nature, and people have actually modified it, made little changes here and there, and made it glow brighter or have a different color. So it was green fluorescent protein, and now there's red fluorescent protein, there's blue fluorescent protein. And those are all done by just making small changes in the DNA and seeing what happens. Right. Yeah. So if they're not like following safety rules or something, they can get kicked out of the lab, and in very rare cases, in like really, yeah, people who everyone would agree that these people should not be. It's mostly people who don't know what they're doing, and they're not really following the safety protocols, and making mistakes. So, yeah, another. Yeah. Not as far as I know, but I mean, you might. So protein modification is not that easy. So an amateur wouldn't be able to really do it, and somebody is well resourced, they can do it other ways. So, but yeah, it is. It might be theoretically possible to make little alterations in the gene, but I think for an overall sequence, they can figure out that it's 90% similar. So hey, you're probably trying to make this, you know. So you just get the CRISPR plasmid, create a guide RNA with the sequence that you want to be cut, and then you have to do a transformation to insert that plasmid into, doing it in bacteria is not that great of an idea, because bacteria anyway have homology-directed repair, in which if you basically put a strand of DNA with a matching flanking sequence, it'll incorporate it into the bacteria's chromosome with that flanking sequence, wherever that flanking sequence match. So you can, yeah. Yeah, so if you make a novel compound or like a biosensor that might have a use. So, there are people working on different ideas, like, GMO, maybe a plant that is resistant to some particular condition. Maybe it can grow in like the desert because you've given it, the ability to retain water efficiently. Yeah. So for small sequences, sometimes you might be needing it for something. I wouldn't. I don't know if it's unethical. It might be useless to just put in a sequence that you came up with. But if you're intelligent about it and you looked at the structure of the protein and you're saying you want to modify this particular part of the protein because you know it contains a motif that you're familiar with, like it might have like a RUV-C, which is like an endonuclease motif, and you want to eliminate that motif, or you can basically modify it and put it in and create it that way. You had a question? Yeah. Um, so doing an agrobacterium transformation, it's not too involved. It would take hours of your time, but then preparing the experiment might take more time. But then you have to wait for the plant to grow if you're doing it to the germline. But if you're just changing, say, part of the plant, like maybe the leaf, you can use a thing called a gene gun, which is expensive if you buy it, but you can actually build one for about $80, and it can actually shoot DNA into the plant cell, and then it'll. So if you want the leaf to be glowing, you just create this plasmid that contains the GFP or whatever, and then you shoot it with the gene gun. You could do it by eyesight, I guess. Yeah, yeah, yeah. It just basically has a CO2 cartridge that, of you have your DNA on small beads, and it just shoots the beads through the cell wall. Some of them go right through. Some of them might bounce off. I don't know, but as long as a few get in, you're okay. So, yeah. All right. Okay. To what extent? Because if it's just creating one protein or something, yes. But making like a plant that has like limbs or something, that might be difficult. Because we still can't even regenerate limbs on the same organism. So these things are not that easy. They, like jellyfish gene, like that's the green fluorescent protein. Yeah, so that is just one gene, one protein. So it it's like saying, oh, I don't want tomato genes in my corn, right? But then you eat corn and tomatoes at the same time, and it's not killing you. So yeah, it's slightly different example, but yeah, yeah. Stigma of. Oh, I see. Like, right. Yeah, right. Yeah, so I think your question is like when we say we're biohackers, does the general public, see us negatively because of the negative connotations that might be attached to the word hacker? Right. Yeah. Right. Um, to some extent, but it's uh, yeah, it's same as when you say you're a hacker, kind of like people might see it negatively, but that's why a lot of people try to say they're citizen scientists or doing practicing DIY biology. So that might have a more positive frame. So if you want to tell someone that you're a biohacker and you think that they might not like that term, you can say, I'm a citizen scientist. So, right. Yeah, so people have this negative stigma about, you're changing the genes of an organism, but actually we've been manipulating life or organisms for like thousands of years. Like I showed the corn plant, so that's not the only example. There's rice, there's domesticated animals. Basically, we've made them dependent on humans because of farming and artificial selection and hybridization. So I think you have another question. Okay, I think okay. So I'll answer it in another place.
I think
Yeah,
so. Thank you, Johan. Thank
yeah.
you
Thanks.
for